Okay, welcome everyone. This is the Humanities and STEM student talk of the 6th Polygent Symposium of Rising Scholars. My name is Maria Dresser, and I'm a grad student in the physics department at Harvard University. I will be moderating today's talk, and the goal of the symposium is to celebrate and showcase the hard work of our Polygent scholars, all of whom have tirelessly worked on their projects over the past couple of months. We have 3 student presenters presenting on a variety of topics today. A fellow Polygent's mentor will be scoring each student's presentation, which will help us determine prize winners after the event. Please feel free to type any questions you have in the chat for each speaker, and after each person presents, we will go through those questions and answer them. Today's speakers are Isabel, Youssef, and Ramya, and we'll go ahead and get started with Isabel's presentation.
All right, thank you. And let me share screen. Okay, so hi, my name is Isabel, and my project is how physical tours impact college decision-making and student success. Colleges offer many opportunities to discover and learn about their school, including researching schools online and through virtual information sessions, as well as seeing the campus through summer opportunities or tours. Other factors in college decisions can include test scores and affordability, though many schools are attempting to be more inclusive. Through every step of the process, colleges are advertising themselves and trying to bring in as many applications as possible so that they have their pick of students. So that brings the question of how to remain objective. Based on past research, a college tour can make a big impression on students. According to a 2019 study, such impressions especially affect students of color who might or might not feel welcome at a school. From the perspective of schools, hoping to recruit students, tours can help less known schools jump to the top of a student's list. Interestingly, taking college courses at a school doesn't have this effect, though it can improve opportunities for certain demographics of students. Confirmation bias will filter any information discovered after the tour or other opportunities to form opinions about the college through the opinions that the student has formed, making the power and effect of these decisions exacerbated. So as someone who's going through this process as a high school junior, as I was touring over the summer, I realized that I drew quick conclusions about what a college was like and whether it was a good fit, especially if I got to talk to students. Furthermore, with the pandemic, I'm very aware that not many students had the opportunity to tour colleges at all, which has challenged the significance of tours. I became interested in the influence of all the components of the college process and especially tours on students' final decisions. Though it is known that tours can affect decisions, I specifically hope to investigate what type of effect that they have, such as what makes a tour good or bad, and how accurate the impression formed is. One potential impact of this experiment would be to help students understand their biases and maybe even counteract them. I plan to send out a survey to colleges to be filled out by their students and hope that the data could help students find a good fit school. So for the experiment that I ran, my first step was to create the survey. I used Google Forms and edited the questions of the survey to be short so that participants would respond and consistent to avoid extra bias and variables. I hope to look at demographic information and prior bias to help process the data, considering the effect of several different factors. And then I asked questions about experiences during the tour, like weather, the amiability of the tour guides, and more. So that was the main data section to find out whether the student was comfortable and happy on the tour. Then I asked questions about the final decision made and whether students felt like the tour affected their decision. The image shows some example questions, and many were in this format of good, very good, bad, very bad, etc. So that was part of the aim for consistency. But some, like the question in the bottom, did not fit this format and were edited so that they would just be as consistent in language as possible. Okay, for sending the survey, once the survey was written, I sent it to 10 colleges and I tried to get a good variety of public and private, large and small schools, etc., so that there was a diversity of responses. I sent to schools such as U Chicago, Rice, and UC San Diego. The email notes that I'm a high school junior interested in decision-making and willing to share results. And for this survey, I directed it to the psychology department as psychology research to distribute to students at the university. I got a lot of data from UC San Diego, but I would love to get some more data from other schools in the future as a next step. Nonetheless, this was a good start and I could already start to see some trends in the data that I received. It could be interesting to expand target audiences in the future. And the graph shows the number of responses from the different colleges. Okay. So in terms of demographics, I got responses from all kinds of people, with interestingly, slightly more female juniors than other demographics. Around 70% of students who toured were expecting to like the school. Some other interesting results include that, as expected from the background research, it is rare to go to a college that you didn't tour if you toured colleges. Students also seemed pretty happy at the schools that they chose and had fairly good touring experiences. Tour guides were considered kind and informative, and students studious and likable. The campus was considered attractive, safe, and fun. Furthermore, over 50% of students were satisfied with their choice. Most students walked away with a better opinion of the school after touring. And this graph speaks to some of the students who might not have been able to tour. So in terms of the interests of groups who toured colleges that they didn't attend, and those who didn't tour, the most important values were academics, majors, location, and price. However, some also valued accommodations, and sports was usually rated as a low priority. Those who didn't tour also seemed more or less satisfied with their choice. For instance, unlike those who did tour, none said that they were unsatisfied, but that was probably due to the small population size. Those who didn't tour were worried about the time and cost to travel far, as you can see in the graph in the top left. They also might not have seen the point of touring or were sidetracked by COVID. So overall, tours seem to improve a student's opinion. And although I would love to get more data, it already seems like we learned some really interesting stuff about how trends show that students tour schools that they expect to like. The tour improves their perception of the school, and then they're likely to attend and enjoy learning at that school. So that's great news for students, as well as the future of college tours. Thank you.
Thank you, Isabel. That was wonderful. So looking at the Q&A, it looks like we don't have any questions posted so far, but if anyone wants to ask questions now, yeah, go for it.
I definitely have a question. Yeah, I was wondering, sort of, as you were doing this research, how did it affect the way you approached your own college visits? And was it strange to like be going through the tours and that process at the same time as doing the research? And yeah, I'm just curious how it affected your own attitudes.
Yeah, so this definitely, touring inspired me to do this project. And I haven't gone on too many tours since, but it is definitely a different lens to look at tours through, and shows the importance of really paying attention to every detail of a school that you get to see on a tour.
Awesome. Thanks for sharing, Isabel.
All right.
If anyone else has questions, please feel free to jump in or post them. I'll wait another minute or so maybe, and then we.
Oh, it looks like I have a question. How did I decide whom to survey? So I sent an email to the psychology departments of several different colleges, and trying to get different types of colleges on that list of people who I was emailing. And I told them to share the survey with students. So that was my process for now, but I'm definitely looking at ways to get this to a larger group of people.
Great, thank you so much, Isabel. Youssef, if you would like to share your screen and start your presentation.
Absolutely. Can you guys see that?
Yes, we can see.
Perfect. All right. Hello guys. Today I'm going to be talking about my project that I worked at, which is called Automelter. Okay, so I'm gonna first talk about the problem that I was trying to solve, and then I'm gonna explain what Automelter is. But basically, a few years ago when we moved to New Jersey, it snowed a lot here, and because I was the youngest one in my family, I was always the one who had to shovel snow off of my driveway, and I really didn't enjoy shoveling snow. I don't think anyone really does. So the three options that I had instead of just shoveling snow over the driveway was, the first option was using a normal shovel, and then the second option was getting an electric snow plow, and the last option was having a heated driveway. And none of these, when I was evaluating these options, none of them actually worked for me. Shoveling snow was cost efficient. Getting a shovel is really inexpensive, but it was really, really time consuming and tiresome, and I just didn't enjoy doing it. The second feasible option was getting an electric snowplow, which is basically, if you're not from the East Coast, it's basically just a lawn mower, except it moves the snow instead of moving grass out of the way. And these are time efficient, but they're super expensive. And the last option was having a heated driveway, which is again super time efficient but really expensive. Now the project I worked on, which solves all these problems, it kind of like gathers the two good parts, which is it is really time efficient and also really inexpensive. Here's kind of like a table that explains the range that we're working with here. So my project was really close to the price of a shovel. A really high quality shovel would go around 50, $60, but it would save you as much time as a heated driveway would. So this is what it looked like, and I'm gonna explain the components later on. But yeah, this is what my initial prototype looked like in my garage. Now, another reason why it could be important on a bigger scale is because according to the U.S. Department of Transport, every year there's around 2,000 deaths and 140,000 injuries happen due to icy roads. And the reason for that is because gritters, and again, if you don't live on the East Coast, a gritter is basically a salt truck. It's a truck that moves around after a snowstorm, and it just sprays salt on the streets, and that the salt helps melt the snow, so it kind of like helps prevent accidents after snowstorms. But obviously it's not the best at doing that, because gritters were invented in the 1940s, and they haven't been updated ever since. Gritters cost a lot to manufacture. They got like, actually 100,000 is really on the low end, and they're only seasonal. And the problem with snow salt trucks is they start clearing the snow off of the driveway, but they don't start until the snowstorm starts with like an hour, or like as it ends. They don't actually start going out as the snow starts falling initially. So the fact that they're sequential, not simultaneous, that is a problem on its own, and it's a lot of research was done on that, and that actually was the reason that all of these injuries were happening. Um, and Soul trucks are also kind of like small moving factories. They're not just a vehicle. There's a lot of machines on these trucks, so they do release a lot of greenhouse gases. Now, another reason Automelter is I worked on this project is because after doing some research, I found out that around 1,000 elders die a year from heart attacks that they get while they're shoveling snow for the driveways. Um, so this is what a street model would look like. So if we could eliminate gritters or snow trucks, and we apply the project that I'm working on a larger scale, so the way it would look is it would just look like a water tower that would have a specific solution that I'm going to be talking about later on, and this solution would be just sprayed on the side of the roads from like tubes, kind of like over here. It's kind of like the same concept. Um, this would do it automatically, instantaneously, as soon as the snow starts falling on the street. So it kind of eliminates the problem where, oh, you need to start later on after the snow actually starts falling, with like an hour or before, or right before the snowstorm ends. So what exactly is AutoMelter? Like, what exactly is this device? So it is a device that sprays a solution automatically as soon as it senses snow. So the solution it sprays is composed of ethanol, sodium stearate, potassium chloride, and water. And the reason I chose these is because it drops the it's a solution that prevents snow from sticking and making like bigger structures, and it also is super good. Like it has no negative impact on the environment. um so I didn't mention this earlier, but salt trucks, when they release the snow, it ends up piling up on the side of the roads and gets washed out in the grass and such, and this is really bad for the environment. It's really bad for animals. Again, there's a lot of research that's been done on that, but this solution, compared to that, is a lot better for the environment. um, so the way it works is there's a temperature sensor and a precipitate sensor that works simultaneously. And what happens is when the temperature sensor senses that the temperature is lower than 0 degrees Celsius, and the precipitate sensor senses that there's precipitate, then they're like, oh, well, the precipitate has to be snow because it's lower than 0 degrees C. So then it knows that there's snow. And then the sensor send a signal to an Arduino. An Arduino is basically just a small computer. And the Arduino sends a signal to the electric valve, which lets it open, and then the solution is just like being sprayed out of the um, gets sprayed out of the tube, out of the container, and through the tubes. And so I'm going to be talking about the four subsystems of the device, which is basically the electronics, the coding, the build itself, and an additional mobile app that I built. So these are the electronics here. So the way it works is, so here on the bottom right, this is the temperature sensor. On the top, the thing with the multiple lines, that is the precipitate sensor. And then it sends the signal to the Arduino, and then it sends it to the relay. A relay is basically an on-off switch, and that is what tells the valve to open or close. There's also on the top right, that is a water boiler, or a water heater, and that is in the container that contains the solution. It makes the solution warmer, so when it leaves the system, it aids with preventing snow from sticking. So the solution is really chemically, it drops the freezing point of water, but it also is warm already, so it just makes things a lot easier. So this is the code flow chart. So the way it works is, if the water temperature is less than 20 degrees Celsius, then the water heater will turn off, because we don't want the water to be too hot. There's no reason for that. That just uses energy, that uses excessive amount of energy for no reason. And we don't want the water to be lower than 0 degrees Celsius. So if it's lower than 0 degrees Celsius, then the heater turns on. And if it's more than 20 degrees Celsius, it turns off. Alternatively, if you use the mobile app, you can manually turn on or off regardless of what the temperature is. So you kind of override the automatic command that is written in the code. The other side of the flow chart is talking about the valve itself. So the first part is talking about the boiler control. This one is talking about the valve control. So again, this works exactly like I talked before. So if you input a temperature and a precipitate condition, and if both conditions are met where it's lower than 0 degrees C and there's a precipitate, then it has to be snow. So the valve turns on, and if not, then the valve is off. Now, when it turns on, it only turns on for like 15 minutes each hour, and that prevents the entire solution from spilling right away for in like the first hour of a snowstorm. So it preserves the solution. So it would spray for 15 minutes and then shut off for 45 minutes and then turn on again for 15 minutes. And it would keep doing that just to preserve the solution. It's kind of inefficient to spray it all at once. Um, and then again, you can overwrite this code by just using the mobile app, and then you would just turn the switch on, and it would keep the valve turned on for as long as you want. And you can turn the switch off, and it would just turn it off even if the conditions are met. So it is automatic. If you do want to treat it manually, you can do that. It would overwrite all the sensors and such. Now here's the build. So on the top left, this is the electric valve. It's connected to the Arduino, as you can see from these two wires. Bottom left, this is kind of the size of the holes, and they're placed around 5-ish centimeters apart. And yeah, it just like placed in a T configuration, as you guys can see here. This just makes it easier for the water pressure to be equal through all holes. And this is the mobile app, super, super simple. It's literally just an on-off switch. On the left here, this is the module that goes in the Arduino, which makes it connect to the Wi-Fi, and then you can connect your phone to the Arduino itself. And literally by clicking on, you turn the water boiler on and you open the valve. Again, this overrides the conditions that's in the code. So if you know that a snowstorm is only going to be like an hour long and you don't really care about preserving the solution for too long, it's only convenient if you know a snowstorm is going to be going on for like 10-15 hours. Then you want to preserve it. But if you know a snowstorm is only going to last for like 2 hours, and you just want to turn the valve for the entirety of the time, you can just turn it on and it will override the sensors. All right. Thank you so much. This is all I had to say. Yeah, if you have any questions, please feel free to ask.
Thank you, Youssef. That was wonderful. I actually had a question because I like to write code myself. So I was just curious, what is this code written or is it envisioned? And if it is already written, I'm curious what language you're using.
It is already written, and I initially actually had it as a part of the presentation, but I took it off because I thought it would be distracting. I thought a flowchart would be easier for people to visualize. So because this is an Arduino, Arduino has its own language. It has an Arduino IDE, which is basically really similar to C++.
Okay,
Mm-hmm.
Great. There is a question in the chat. How do you get this in the hands of more people? What's your next step for scaling it up now that you have a working prototype?
How do I get this in the hands of more people? So I've built my second prototype just a few months ago, and I have been talking to a few manufacturers. I've been trying to get a patent and stuff like that. So I'm really trying to get it out there as soon as possible. I'm trying to save a few lives before I can move on with my life. But yeah, I think it would make people's lives convenient. It would save a few lives, even if it's like one or two out of like the thousands. That would also be nice. What are your next steps for scaling it up? So so far I've talked to a few professors, a few people who are in the field, and they thought that building a prototype for driveways and having it be a product is extremely doable. But scaling it up to be for street use and replacing gridders or salt trucks would be really difficult, just because this would be a big infrastructure move, and I would have to convince a whole city to move away from salt trucks into this infrastructure thing that would cost a lot of money to build initially. But operating it over the long term would actually save a lot of money and save the environment as well. But yeah, the second one is a lot less feasible, I would say.
Great, thank you. There's another question in the chat for you, I think from Isabel. I'm interested in whether you can change the amount of solution sprayed with the type of valve you used.
Yes, so actually the valve is adjustable and I could have done this in the code. I didn't, but like now that people think about stuff like that, maybe I could add it to the code and have people adjust the amount that's flowing out. The only reason I didn't do that is because the amount flowing out naturally, like the pressure of the solution flowing out naturally decreases because there's less water in the tank. There's less of the solution in the tank, so there's like less stuff pushing down on the solution, which is why I kind of like didn't really consider doing this. But adding a pump or just adjusting the flow rate of the valve is really doable. So that's something I could keep in mind. What is the most difficult obstacle you face during your projects? Okay, actually all of them, the entire freaking project. So when I started working on this, I had absolutely no idea how to code, like zero. This was my first time ever coding in my life. I have never worked with electrical components. I've never worked with Arduinos, never touched like wires before. Um, the only thing that I kind of had experience with is actually building stuff physically. But other than that, like everything else was kind of new to me, and I learned a lot. I learned a lot of skills with coding and electronics through this project. So it was definitely a learning curve, but it's something that I really enjoyed. So it wasn't really an obstacle where you just face it and you're like, ah, now I'm bummed out. It was a really enjoyable learning curve because it was something that I was passionate about.
Thank you so much. Okay, great. I think those are all the questions I've seen so far. So Ramya, if you want to share a screen and start your presentation.
Okay, yeah. Can you see my slides?
Yes.
Okay, great. Okay, hi everyone. My name is Ramya, and today I'm going to be sharing a little bit about my polygens project that I did. It's titled The Efficacy and Impact of CRISPR Repression of the ERO1A gene in Hepatocellular Carcinoma. And during this project, my mentor was Melanie. So I know that was a bit of a mouthful, so I'm just going to break down each of those parts of my project. So first of all, I wanted to talk a little bit about what hepatocellular carcinoma is. So hepatocellular carcinoma is the most common form of liver cancer, and it is especially common in patients with chronic liver diseases. Those who have hepatitis B or C are also more likely to have or develop hepatocellular carcinoma. Though it is treatable if it's discovered early, it usually isn't discovered until much later, at which the prognosis is only 6 to 20 months. So it was a disease that I was really fascinated by, and that's why I wanted to investigate it more in my project. And there are several treatment options that are being tested for hepatocellular carcinoma, like surgery and implantation and gene therapy. But one I was specifically interested in was CRISPR gene editing. So CRISPR gene editing, like you might know, has greatly impacted the field of biomedicine since its discovery in 2012. It is often an effective and accurate way of targeting and altering a specific portion of the human genome. There's a lot of research and innovation that's underway for its therapeutic applications in genetic diseases, including cancer. So I looked at CRISPR gene editing that specifically targeted the ERO1A gene in hepatocellular carcinoma patients. So the ERO1A gene stands for the endoplasmic reticulum oxyreductase 1 alpha gene. And like its name suggests, it is found in the endoplasmic reticulum, and it codes for the ERO1A protein. So the ERO1A protein is involved in the formation of disulfide bonds along with disulfide isomerase, and these are needed for the proper formation and conformation of proteins. It is being implicated in many diseases, including cholera and liver cancer. So in liver cancer, like HCC, its overexpression leads to tumor progression. It inhibits the T cell response in partnership with polymorphonuclear MDSCs. And while the CRISPR edit specifically targeted the ER01A gene, I was curious to see whether this edit could have possibly impacted other genes that are related to HCC as well, to see whether the patient's overall health could be improved by this method. So one specific gene I investigated was the UPF1 gene. It is traditionally underexpressed in HCC, and it is involved in nonsense-mediated mRNA decay. Nonsense-mediated mRNA decay is important to eliminate premature stop codons. However, since it is underexpressed in HCC, there is NMD deficiency for HCC patients. This leads to the disruption of the immune system and induces the epithelial mesenchymal transition and promotes HCC growth. There are also certain genes that are overexpressed in HCC that I wanted to investigate, including SPP1. So downregulating SPP1 has been found to promote cell apoptosis, which is important in the immune system. The ARG1 gene also suppresses the T cell proliferation and the synthesis of cytokines, and then the MDM2 gene leads to the inactivation of the p53 protein, which can impact the pathways that are related to the p53 protein as well. There are also two potential biomarkers for HCC that I investigated, including GPC3, which forms complexes with WNT. WNT signaling maintains cancer stem cells and progression of tumors. So this overexpression of GPC3 in HCC was something I was curious to investigate. I also looked at KPNA2, which is involved with the nuclear export of proteins, but its exact role in HCC is not well known yet. It is transcriptionally repressed by the p53 protein as well. So I want to go a little bit into my methods and materials now. So since I performed this project during COVID, I was not able to go to the lab to physically do the experiments, but rather how I obtained this data was through an online database called ENCODE. So the University of Connecticut performed this CRISPR editing in vitro to repress the ER01A gene in a male Caucasian child with HCC in their 2019 trial. And then they had published this data publicly on ENCODE, which is where I was able to retrieve it. And therefore, I was able to perform RNA-seq analysis to study its effects on the gene expression of the genes I just discussed. I just wanted to mention that this project is not affiliated with the University of Connecticut. It was just something I was curious to explore. Like I just mentioned, I retrieved the data from ENCODE in the FASTQ format. I then performed quality control measures just to make sure that the data was to the appropriate quality that I wanted for my project. Then I performed alignment and quantification using Callisto, and I used Digus to visualize my data and its results at the end. So now I'm going to dive into my results. So first, we can look at the differential gene expression log fold changes for the ER01A gene. We see here that it is negative 2.598, and also the counts for transcript abundance for the modified sample are significantly lower than the counts for the control, which all indicate that the gene expression has successfully been decreased with the CRISPR gene editing. And this indicates a decrease in the tumor metastasis and migration, which was super interesting to see. I also want to talk about the two biomarkers that I investigated, GPC3 and KPNA2. So we can see by the negative log fold changes that its differential gene expressions have also decreased, and the counts for transcript abundance for the modified sample are lower than the control as well. We can conclude that this would boost anti-tumor immunity, but since both of their roles in HCC is not too well known yet, there is a need for further studies to confirm and solidify this prediction. And the third table is what was most interesting for me, which was seeing how this CRISPR edit actually impacted all the other genes that are related to HCC as well. So the first being UPF1, which I mentioned before, that is traditionally underexpressed in HCC, but with this CRISPR edit, we see that the differential gene expression log fold change has increased, which would indicate a more efficient NMD pathway. to eliminate the premature stop codons, like I mentioned in the background. And then for the SPP1, ARG1, and MDM2, I talked about how they were all overexpressed in HCC. But as we can see, due to the negative numbers in the log fold changes, as well as the lower counts for transcript abundance for modified samples, we can see that all of their differential gene expressions have been decreased with the CRISPR edit. For SPP1, this means that it would better impair the proliferation of HepG2 cells in HCC. The T cell response will also be enriched with the ARG1 differential gene expression decreased, and the P53 activation will increase, and this will promote the P53 related pathways. So I just wanted to restate some of my takeaways from this project. I noticed how the CRISPR gene editing was successful in repressing the ER01A gene. It also decreased the expression of potential HCC biomarkers like GPC3 and KPNA2. And we see that it also surprisingly decreased the expression of SPP1, ARG1, and MDM2, which are all related to HCC, and increased the expression of UPF1. Though there was only one available sample for this experiment, indicating it might not be statistically significant, it still helps us to understand the potential of CRISPR gene editing in diseases like HCC. Thank you. I'd love to take any questions.
Thank you, Ramya. That was very impressive. Yeah, everyone could just feel free if you want to unmute yourself and ask, or send it in the chat for questions.
I have a question. What do you think is the most promising next application of CRISPR?
I think like the next promising step is probably like personalized medicine, like seeing how we can personalize it to each person's specific like history. And I think that's like really interesting prospects. And next year, I hope to study more about CRISPR when I attend Johns Hopkins University as well.
Congrats on your acceptance.
Congratulations.
Yeah, thank you.
My question was, of all the different, I guess, cancers that you could be looking at, and I know, I mean, I'm not a biologist, but I know that CRISPR is something that is investigated in a lot of different diseases and how it can help or hinder. And I was wondering, how did you pick this particular liver cancer to work on?
Yeah, I think like there's two parts to it. The first was like I mentioned, HCC is like there's still a lot of like discovery and research that's underway for it, since it's not very well known, and there's also a limited prognosis for patients when it's discovered later. So that was very appealing for me. And the second, I guess, is also just the limitations that exist when you perform research virtually, because I wasn't able to conduct it hands-on. So I kind of relied on like what types of data sets were available for me to work with on ENCODE as well. And out of all the data sets I looked at, this one was really promising for me and really interesting. So that's why I chose this.
Very cool. Thanks for your explanation.
There's also another question I see in the chat. What was the most surprising, unexpected result of your research?
I think like what was really surprising for me is seeing like how many other genes in HCC were affected because of this CRISPR edit as well. Like the CRISPR edit targeted the ER01A gene, but still we see how the UPF1 and like SPP1, they're all like changing in their differential gene expression. So it's really interesting to see how even if we only target one gene, there's still a lot of impacts and implications. So it was really interesting just to see the other genes that are related to HCC being impacted as well, because I didn't think about that initially. And then I also see another question about what I would have done differently if I could work hands-on. I think if I could work hands-on, I would have been able to probably look at more like samples. I was only able to look at one sample since that was the only one that was published online. I would like to conduct it on like more patients and more samples, because that will allow for the results to be more accurate. I think working hands-on would have also helped me to learn more about like the lab techniques that are associated with this, which is also something I'm very curious about, especially in research. So I would really look forward to doing hands-on research next year, hopefully, if the labs are more open and welcoming.
I just want to say to all of the presenters that I am very impressed, and it's been an absolute joy to see all of your work. I definitely didn't do anything this cool in high school, so you should be very proud of yourselves.
Thanks, everyone. Congratulations. You guys all did amazingly.
Fascinating presentations and work. So we have a few more minutes. I'm curious, what skills you learned that you think will be the most valuable moving forward? And I'd love, like, if we have a few more minutes, Maria, if we could, like, I'd love to hear from all of the presenters today.
I think I can go first. Like for me, there was a steep like learning curve initially. Like I think some of other panelists also mentioned, but there was a lot of things we didn't know about our project or like the techniques on how to do it going in. But it was just like really rewarding to see how I could still learn all of these things in such a short time and apply it during my project. My project, I think, only was around like 2 or 3 months or 2.5 months. So it wasn't too long, but I was still able to grasp things really quickly. So I learned how to sort of learn on my own, but also like ask my mentor for help once in a while. I kind of gained a sense of confidence in myself that I could like pick up on things quickly. And even if I didn't know something, that's okay. I could still learn and still try. So I really like motivated me to continue like doing research and understanding that it's okay to not know everything in the beginning, but that we can still like work together and find resources online to learn about things.
Yeah, and I can go next. I think that I learned a lot about perseverance because it was hard to get responses, but also about how to write a survey. It was actually quite interesting to think about the psychology of asking these questions and how a subtle change in wording might influence the way that somebody answers the question. And so it's not like you just write what you want to ask and then send the survey, but actually I realized that there are all these other factors going on in survey writing.
I learned how bad things can go when you have a bajillion components that you're working at and a lot of different parts of your project. I remember there were like a few weeks throughout my project where my mentor and I, like my project just straight up wouldn't work. Like something would go wrong, and it would take us a few weeks to figure out. Obviously, like the fact that we were working like online made things a little bit harder, but like we would look at the code and then we would look at all the electrical components and make sure everything's connected correctly, and we just take like look at each thing individually for like 10 minutes and figure out what's wrong, and we just wouldn't figure it out. And like after 2 weeks you look at the code again and just like see if we forgot like a semicolon or something. So yeah, I think like attention to detail, especially when you're working with something that has a lot of parts and organization, is something that I learned. I also thought that I'm majoring in mechanical engineering, and I thought that okay, if you have a good plan and you have all the components, just put everything together, slap it on. It's probably gonna work if you put everything in correctly. But no, that doesn't work because you're never always gonna put everything correctly where it belongs. If you have a lot of parts, and I just finished an electronics class where we build circuits and things like that, and it's also again the same exact thing. Like no matter how correctly you think you placed things, you need to look at it like 100 different times and then you'll find something wrong with it. So yeah, I think it taught me patience and attention to detail. But yeah.
Love those responses, and so amazing to hear some of the recurring themes around perseverance and continuing despite setbacks, which is very much the theme, it seems, based on my experience and everyone else's, of doing research. Well, I really enjoy this. Thank you so much.
Yeah, I wasn't sure if we should end earlier. I don't know. I actually did just come up with one question though. I was just curious to hear like what are you all juniors or seniors or like what year you are in, and then if you have like a after high school goal. I'm just curious for each of you what year where you're going.
I can go first. So I'm a freshman studying mechanical engineering at Northwestern University. Um, it's kind of weird that I'm the only college freshman here, but it's because I worked on this project like towards the end of my senior year. Um, yeah, that's why I'm here right now. And big goal projects probably do some cool aerospace stuff. I want to build rockets. I want to help like put a man on Mars or something. I don't know. I think that would be cool. But
And
yeah,
what advice do you have for incoming freshmen like Ramya
incoming freshman
into college?
into college, definitely Google Calendar. Google Calendar is your best friend. It's definitely underrated. I see a lot of my friends, they just try to remember like stuff that they have or like post-it notes. Uh, Google Calendar, because it's always on your phone, it's on your computer. 100% Google Calendar is your best friend. It's the way to succeed. And generally just time management, which comes from Google Calendar. It's all about time management in college. College actually isn't bad once you know how to manage your time.
I can go next. I'm Ramya. Or I already said that, but yeah. So I am a senior in high school right now, and then next year I'm going to go to Johns Hopkins University, and I'm going to be studying biomedical engineering. So I hope to work a lot more with like CRISPR and like more on medical devices and imaging too.
All right. And I'm Isabel. I'm a junior, so still just starting the college process. And I am, I guess, interested right now in a little bit psychology, but also physics. So, Maria, if you have any advice, I'm taking that class this year, and it's really interesting to me because I love math. So,
I think physics is the best thing in the world. So that's what I have to say. It's very hard, but I think that's the joy of it. So if you like a good challenge, physics is a great place to go. I guess that's my advice. Just probably stick with it. When I first started physics, I actually thought I hated it because it was so hard. So if it gets really, really hard and you feel like you want to give up, maybe try to like push a little bit more and see if you get over that hump. And then it becomes very rewarding.
Yeah,
Maria, can you, oh, sorry. Youssef, you go first.
I was considering an astrophysics major before I settled down on mechanical engineering, and then I looked at all of the class, and I was like, "Yeah, this is a little bit too theoretical for me. Like, if I can't see or build something, then it doesn't exist for me."
I was just about to say, Maria, can you say a little bit more about your own work, actually? I'd be curious to hear what projects you're working on and what your dissertation will be about.
Yeah, of course. So my project is a little more applied than like some physics grad students might be working on. So I do computational physics, and I work on designing new materials that can be used in batteries. So like as I'm sure you know, batteries are very important in terms of adopting renewable energy because like solar and wind power is intermittent, and so it's important for us to have like a storage for that energy whenever the sun goes down or the wind's not blowing. We want to be able to store that excess energy. And my particular research basically looks at, I work really closely with experimentalists who are actually building the batteries. And so sometimes they will come to me and they'll say, hey, my battery is having this problem. I don't really understand why. And so I will do the research on the particular material, and I work on figuring out how we could change this material to make it work better in the battery. So maybe we want to dope it, or maybe we want to grow it so that it has a slightly different crystal structure. And I look at all these different types of materials and compare them to each other to see like what would be best in a given battery.
So cool. Thanks for sharing.
What did you do your undergrad in? I'm kind of curious because I studied a lot of these things, and it was in one of my material science classes. So, like material scientists and material engineers usually do a lot of that work. So I'm kind of curious.
So my background is actually, I did a double major in math and physics. So I had never really learned much material science until I got into grad school. And now I'm taking more material science classes and learning a lot and realizing that material science is super fascinating.
Right?
Yeah, it's great. I love it. Well, I guess we are finally at time, but unless anyone has any last questions or comments. All right. Well, thank you everyone so much. Great
Thanks,
work.
everyone. Have a great
Thank
rest of your
you.
Saturday.
Bye.